Hello, everybody. Welcome. We're excited to have you. We're going to give it a couple more minutes here as we wait for everyone to join us for this webinar on microbial nutrition in citrus agriculture. Really excited conversation we have going on today with Herb Young, and we will just give it a couple more minutes here and see who else is going to join us. Excited to have everybody. Hello, hello. And for those of you who are joining us virtually today, if you miss anything or have to hop off, we will have this recorded for all of our attendees. So do not worry. We'll have some great discussions at the end. Just give it one more minute here before we get started.
Marie, maybe 10 minutes discussion at the end? Is that what we're shooting for?
Yep, 10 or 15 minutes.
Great.
We'll have some time for Q&A at the end, so we'll frame all that up here in just a minute.
That'd be great. Looking forward to it. Yeah.
Yeah. Awesome. Well, we have about 87 folks with us today, and folks will stream in as we go, but we can go ahead and get started. A few housekeeping items before we dive into the amazing content we have prepared for you all today. If you have questions that you would like us to answer, we will be saving time at the end for some open discussion. Any questions that we don't get to, we will try to respond to you and get back to you in the next week here. But for the questions, we'll have some time to dive into that. So please engage in discussion, ask questions. Herb is a expert here, and we've got Gus and myself, and we'll make introductions here shortly, but just want to let you all know that you can post questions in the webinar. And we're excited to have everyone here today. So just some really quick intros to, if you haven't interacted with Biome Makers at all, if you haven't interacted with our team, I'll just introduce us really quickly. I'm Marie Mullins. I'm an account manager with Biome Makers. We have Gus and Herb joining us today, and I'll introduce them here shortly. But Biome Makers is a company that is decoding the soil microbiome. So we're taking all the complexities of soil biology and breaking it down into a functional analysis. So as a technology company, we really strive to help both growers and product manufacturers in understanding the microbial interactions in the soil throughout a growing season with a new product application and so on. And so today what we're going to be talking about in that context is how different management practices affect soil ecology and soil biology, and how Herb has taken on his own operation, compared more conventional traditional management practices to what we're calling indigenous management practices, and then regenerative management practices. And we'll talk about what all of that means, but being able to look at those things from a biological level and understand how those things all are connected. So Herb, that is joining us here today, we're really excited to have with us. Herb has over 40 years ago, he came across the concept of agroecology and really drew him to the field of agronomy. His career eventually led him to the pesticide industry with BeCrop Sciences, which he worked, I think, for 35 years, right, Herb?
Yeah, exactly.
Yeah. With his retirement came a decision to farm and grow citrus himself after years working in agronomy. This decision really brought him full circle back to regenerative and biodynamic systems, which he's now really passionate about, and we're excited to learn from him about how he's been restoring the microbial balances in soil and plant ecosystems, his dedication to his work that led him to conduct DNA analysis and really take a deeper dive on his own operation into field trials and understanding things at a deeper level. And he began his growth in 2021, is that right?
Exactly. Yep.
Yeah,
just 2 years.
So he's just getting started in his research farm, but he brings 25 years of field research experience and over 35 years working in agriculture. So we're really excited to have his expertise to chat about and understand what he's got going on in his own operation after so many years working in the field in agriculture. So we're really excited to have you, Herb. And I just want to introduce Gus as well, who's our technical agronomist. We're really excited to have him. He can help us just in diving into some of the portal tools if we need it, and answering any of our technical questions that we have here today. So we're really excited for this discussion. And as I mentioned, just really quickly, Biome Makers, we're a global company. We're really excited to be hosting these webinars regularly to bring more technical knowledge on soil biology to the industry. So we're really grateful for all of you that have joined us today. And yeah, I think Herb, I'm gonna pass it over to you to take us on this journey.
Sounds great. Thank you so much, Marie, for the introduction. This was quite a leap for me after spending a career in the pesticide industry. And to be honest, very sincerely, I think most people in that industry believe in what we're doing, that we're helping farmers and have the ultimate goal of feeding the world. I made an interesting decision when I decided to farm on my own. Being a plant pathologist and having all this huge experience, I just was making a financial decision, and I thought, well, you know, I can grow organically and probably have some financial advantages, and I think I can figure that out. So about 3 years ago, a year before I ever started, I started researching and immediately literally fell into and discovered regenerative agriculture. I was overwhelmed at the amount of research and the incredible detail that was in the regenerative side of agriculture. And I call it conventional agriculture's best kept secret. It's something that we knew nothing about. I had never heard the word regenerative. So I became excited about the possibility of actually trying to do something completely different, restore the microbial ecosystem, and have really taken that, started as a challenge, and it's actually really become a passion. So let me give you a little history of how I got to here. So just a little over 2 years ago, the farm I bought was a cotton farm. And so I had to literally wait for the cotton to be harvested and get off the field. Then the field was realigned to go north and south for trees instead of the alignment. I did the last tillage that this land is ever going to see. I did a deep shank tillage to make sure that there wasn't hard clay pan left over from the cotton farming. And then I started to plant citrus trees. So that was a full 2 years ago. And these are trees now that are just 16 months old from that little sprig that you saw on the back of a gator. My goals completely are a healthy tree that's going to be grown 100% organically and 100% regeneratively. There's a lot of ways to measure that in the tree, and that's what I'm finding out. One of the easiest, quickest is the bricks in the leaf, the amount of dissolved solids that are in the leaf tissue. And it tells me immediately what the nutritional level is in that tree. My trees are actually looking different than the growers that are in this area. I live in South Georgia, which seems kind of unusual for citrus production. A couple of things have changed recently that have allowed citrus to move further north into colder areas. One is cold-resistant rootstocks and the innovation of microjet sprinklers. Every single tree has to have a microjet under it during cold snaps, which is 8 or 10 times a year. The sprinklers come on before anything freezes, and we have to bathe the tree in nice warm groundwater that's 65, 68 degrees, and it keeps the trees alive. So that's where we are. And then the other thing we have in Georgia are more consistent cold winters, which put our trees into dormancy, which Florida doesn't have that luxury of. A big difference between Georgia and Florida, about 150, excuse me, citrus growers in South Georgia now. Amazingly, it started with satsumas because they're the most cold tolerant. And it's expanded now to, we're now working hard to get other varieties to grow. It's all fresh fruit, a little different than the juice industry, which concentrates in Florida. So what about nitrogen? Yeah.
I guess, can you just talk a little bit to what, so I heard that it was kind of an economic decision that, or that was one of the drivers, right? You thought that organic growing in particular could give you a market advantage, higher profit margins, potentially higher value products, right? Coming off of organically produced.
Absolutely, yeah.
Right. So that I heard was one inspiration. Can you talk a little bit about other inspirations that kind of took you down this journey to regenerative specific? Because we don't have to get into too many details, but there are differences between organic and what regenerative really embodies.
Absolutely. The idea of organic just started it. It was a financial decision once I discovered how much research was being done regeneratively, that there was an entire ecosystem under the soil that was allowing the trees to interact with the cover crops. Then the whole idea of doing it to be organic went completely out the window. To me, yeah, that's a good point. Organic, a lot of it is about, to become certified organic, and I'm working to be, it takes 3 years. It's what you don't do. You don't use pesticides, synthetic fertilizers. And, but for me, regenerative is everything that I'm going to do, which is concentrate, literally focusing on the microbes in the soil. And that's where a lot of the new testing procedures, and particularly the Biome Makers, their BeCrop analysis, the DNA in the soil has been absolutely revolutionary. So what started as an idea that maybe it could be economic, that's not even in the picture anymore. Now I'm just really passionate about validating regenerative movement. There's so much good information. A lot of times there's not a lot of hard research. And having done 25 years of field plots, I feel like that's what I can bring, is the chance to, whenever I have a question about what should or shouldn't be done, I usually end up putting out a replicated field trial. I've probably got, right now I've got 8 big replicated trials on my farm, which is not very big. So, but it's been exciting to be able to find some things. So these are some, thanks Marie for getting me back on track, but these are kind of the goals that I have to be completely regenerative. This has moved way beyond organic. So the tree looks different. I'm almost growing a tree focusing on calcium instead of nitrogen. I'm not going for the expedited growth that you get with nitrogen fertilizer. Tree results in nice tight internodes. This high bricks hopefully is going to translate into low insect pressure, better, thicker cell walls, better disease control. Citrus is an interesting crop. It can flush 5 times a year. And you can see this tree on the left. You can get 40, 50% on a young tree, 50% expansion in the tree in a single flush. So this is my goal, trying to get maximum production, maximum growth in the trees simply through organic fertility and regenerative. I feed the microbes in the soil. It's goodness, at least every month. There's quite a concoction. They get seaweed, kelp, fish hydrolysate, and then a real complex mixture of humic acid, fulvic acids, and molasses, which is an extremely long string carbon source, and so some specialized products, and I'll show some of those. So my ultimate goal, back to the reason I'm farming, which all farmers, we have to make a living at it, is now to have nutrient dense fruit and to be able to show that nutrient density is something completely different that comes out of regenerative growing. So one of the biggest ways that I get there is through cover crops. It's completely different look to a grove. A row of citrus has an irrigation tube on top of the ground, so I can't mow between the trees, but I'm growing cover crops. I seed them twice a year, at least 8 or 9, 10 species. Here's some of the ones. For quite a few reasons. The cover crop, when I mow it and blow it over towards the tree, adding all of that nutrition back to the tree, but really also back to the microbe. And I'm developing a nice thick mulch layer on top of the ground, protecting the microbes. And there's been some huge changes just in the soil in the last 2 years. So I wanted to.
Herb, one question I'm sure you get all the time while we're on talking cover crops is,
Sure.
Do you have any concerns about competition from those cover crops with your citrus trees?
You know what? That is the classic ag chem concept. We always taught that cover crops, that weeds would reduce yields by 40%. I mean, that was kind of the excuse. And most everybody grows citrus. They spray with clear ground. I'm depending on all of the research that's saying that there is an interactivity between the cover crops and the tree. My long-term goal is to have mycorrhizal fungi that completely dominate the ground in my soil. Each one of the reason I use so many cover crops, each one has a different exudate profile. So every cover crop in these pictures is feeding the microbes in a different way. I've got legumes, grasses, crucifers, several other things, and I change them every year. So there may be competition, and I'm going to show why the regenerative trees are a little bit slower to take off and grow initially. But I think it is probably a sacrifice that I'm making in the first year, but I really want to establish this relationship. I even went into the nursery before the trees were ever brought out and inoculated them with mycorrhizal fungi. So I wanted to show a couple of the tests, quite a few that I've done, and to see how the BeCrop and the Biome, the DNA interacts. This was a fascinating trial that was presented to me of using in-soil algae, which is Chlorella vulgaris, is the algae species. And you can see these two. So I did a trial of, and everything I do is replicated. So I used, there were 8 trees in a plot and then 4 replications. And I did an untreated, a foliar treatment of the Chlorella vulgaris, and then through the drip injection. The sprinkler head is inside that, it's called a tray teepee, that plastic cone around the base. We leave that there for the first 2 years of tree growth on some trees. So you can tell these 2 trees, completely different leaf flush between the untreated, and on the left is the soil application. So I'm taking data constantly. I took trunk diameter measurements and then leaf flush rating. What percentage of branches had a new flush of leaf coming out the end? And so on this slide, the untreated would be the normal cycle of that tree. So you can tell there were two applications of the in-soil algae, and then the rating started 2.5 weeks after the second. The applications were 3 weeks apart. And so the expected leaf flush was 25, 30, 35% at 2.5, 5, and 8 weeks. The foliar spray was exactly the same. But something happened when that algae, which is hormonally charged, was applied to the soil. We have a complete response, and the tree at 2.5 weeks, the leaf flush jumps to 55%. And so there's a lot of, luckily we had soil measurements to show why this might be happening. So this was the BeCrop results.
I just have one quick question.
Sure.
For those, so we have a diverse audience. I'm sure there's a ton of citrus folks that have joined us, and I know leaf flush is important in a lot of different tree crops, but can you just explain what leaf flush is really quick and why it's important and why this is really neat that we saw this difference?
Yeah, absolutely. So citrus are a different crop. They don't ever drop their leaves, unfortunately, except the bad freeze that we had at Christmas. But so the leaf is the factory. We're trying, although my trees aren't in production yet, I'm trying to build a tree and I want as much leaf canopy as I can possibly get that's going to feed in to grow roots, grow scaffold, grow wood. So for decades, that's always been a citrus grower's goal, is to get additional leaf flushes. There's products been on the market that growers have spent a lot of money just to get, when they're getting a fruit crop coming on, they'll put on products that'll give them one more leaf flush to have that much more production capacity into the tree. So I'm growing dense, tight trees, which are a little bit different. A lot of the competitors around have a spindly tree, not, you know, a third the leaf canopy that I'm trying to grow. So that's kind of my objective, is to grow this intense tree. So here's the BeCrop, the things that I learned. So this was quite a surprise. So it's exactly what I saw above ground. The below ground DNA confirms it. So got a little bit of increase in the number of species in the soil, which those, but the real differences were the soil quality, which BeCrop rolls all the factors together that they're evaluating, knowing what species are there, the quantities of those species. They give an overall soil quality rating. Well, that bumped up on my injection treatment, and so did the biodiversity, and so did, and then when they roll those things together, they've got measurements of the nutrient pathways. So all three, N, P, and K, increased dramatically with the foliar and with the soil treatment. But I saw it was able to measure it above ground, also in the trunk diameter growth. And then here's another measurement that comes out of BeCrop. So this dashboard shows that both of the treatments of N soil cause the carbon to increase, go into the darker blue. The nitrogen is fascinating. So it was reduced, and simultaneously in this trial we did Haney soil analysis, and the Haney test was even more interesting, because all of the nitrate and most of the ammonium in this soil applied N soil was converted to organic nitrogen. So basically, by putting all these tests together and realizing that, seeing the total picture, we've got microbes that are being activated by the algae, and they are consuming all of the nitrate, the ammonium, and they're actually producing amino acids and proteins, providing that to the tree, and then we get this explosive growth. So interesting when you roll it all together. Yeah, go ahead.
Yeah, I was just going to comment a little bit on the indexes here. So, you know, for those of you who are kind of new to Biome Makers and the BeCrop test, what we're looking at here is a tool from our portal where we have, we're looking at the microbial potential or the biological pathways for these different nutrients. And so here we're looking at untreated versus in-soil foliar and in-soil irrigation. So whether it's soil applied or foliar. And in each case, we're seeing, you know, these indexes here from very low to very high. Every pathway was improved in the in-soil applications versus the untreated. So Nitrogen went from very low to low, which could still mean a 20% increase in overall nitrogen cycling, which, like you talked about, Herb, is like the, going from organic or inorganic to organic forms of nitrogen that are more stable, that are going to stay in the soil and have less leaching, and plants can consume amino acids. So we're improving the nitrogen cycle overall, as well as phosphorus and potassium. And the phosphorus and potassium are significantly higher potentially in the, in the in-soil foliar and irrigation application. So just kind of wanted to give a little.
Absolutely. Yeah, it paints, it's so incredible to see what's going on in the soil when you see above ground effects. So
yeah, it is really neat. And I guess it's a really cool point, because as we continue to go, a lot of field trials, and you would know better than anyone with all of your years of experience in field trials in specific, for years we've just looked at yield and or plant vigor or greenness. Like, we've only had mostly above ground metrics to really measure changes in the different programs and products and practices like cover cropping and so on. So adding this microbial layer, we can really start to validate the things that we see above ground, below ground. And I get really excited about that, to add the scientific validation of the stuff we can't see necessarily, but now we can prove, even though we kind of knew it already.
Well, and with biostimulants, biological products, regenerative practices like cover crops, it's very difficult to really see those tangible benefits in using, conventional chemical fertility tests, because a lot of these biostimulants, biologicals, they're not NPK containing products. There are, of course, some out there that contain some combination of nutrients and biostimulant properties, but what these products and management practices like cover crops are doing is affecting the microbiome in ways that improves those nutrient cycling functions we just looked at for carbon, nitrogen, phosphorus, and potassium. And now with BeCrop, we can hone in on that. And rather than kind of drawing assumptions based on metrics like yield and chemical fertility, we can actually measure those changes in the microbiome to see tangible improvements over time from practices like Herb is demonstrating.
Yeah, it's exactly, in that trial, there was no fertility difference, but it was the algae stimulating the microbes that completely changed the nutrient profile. It's really exciting. And to be honest, that's what regenerative agriculture is about. So this is the major trial that I've got. I've got, 135 trees in this. These are, I wanted to evaluate for myself whether regenerative was significantly different than conventional. So my conventional trees, I did as painful as it is, Roundup herbicide strip, synthetic fertilizer, whatever pesticides the growers around me were using, quite a few insecticide sprays and nematicide treatment, a couple of fungicide. But my regenerative are completely organic fertility, and I'll get into that a little bit, and then added microbes. And then I wanted to see what the indigenous microbes are doing, what's already there in my field. And this was my shock when I first did my first BeCrop sample. And I imagined how many microbes are there going to be when I pull a DNA sample out of a naked cotton field that's just been tilled. And I thought, oh, there'll probably be 20 species. Well, there were 680 species right off the bat, indigenous. So that treatment is to see where they're going to go. Now, this is my fertility program. So this is the organic fertility just used in the indigenous and the regenerative treatments. I got some good assistance from AEA the first year, and I do sap analysis. Routinely, it breaks down every single element that's in the leaf. You take a 100 leaf sample and analyze it, and you get the old leaf versus new leaf. And then this chemistry is broken out. So I've literally got a jug of each thing, copper, manganese, iron, zinc. My nitrogen is completely from fish hydrolysate. Carbon is from quite a few different sources. The molasses. So this is feeding the microbes. And then I really had a breakthrough by using this as foliar treatments. And I got some tremendous tree growth once that started. So this is the first copy where I started with BeCrop, and this is the indicator that you get. This is the paper version. In the last year and a half, they've developed a dashboard which has tremendous amount of information, and we'll try to get some time to share that. So this is the BeCrop samples that I've taken. I took a pre-sample before the trees ever went in the ground, April of 21. And then 7 months after the trees have been in the ground, I pulled BeCrop from the 3 replications, consolidated them, completely made sure it was uniform. So that's October 21 is 7 months in the ground. And then early the next summer, I took my second full set from each of the 3 treatments. And then early this coming summer will be my final set of samples. So in the first, so on the left is the pre-treatment, 680 species. This would be fungi and bacteria combined. So once a tree was in the ground, once you still got some roots, that's the key. Even the conventional, the number of species went up. So that's probably awakening of species that have been dormant for a long time. The transitional or the indigenous treatment, I now call it, went up 200 species. And my regenerative, where I literally add microbes, and I use quite a few different sorts of microbes, including mycorrhizal fungi, went up 400 species. So this was my excitement the first year. And then in tree crops, we really concentrate on fungal population in the soil. The ideal is to get it up above a ratio of 1 to 1. Vegetable crops, row crops are bacterial dominated in the soil. Tree crops should be fungally dominated, but I'm not currently. The field was kind of a vacant. From a fungal point of view, every time the field was tilled, it destroyed the fungi that were there. The mycelia would have been 0. And that's my big goal, is to work back. So now that I'm 7 months in, the conventional, which has a Roundup strip, it's bare ground, fungal population drops way off. The transitional indigenous stays about the same. And now I've got this nice increase in the regenerative treatment. In my fungal, this is not fungal species, this time it's detects. How many detects? Which these numbers are amazing. 150,000 in a gram of soil. So I'm now 6 times the more fungal population in the regenerative versus the conventional. And then everything changed when I went to the second year. So this is now the summer treatment, summer evaluation, the second year. And this is the this is the new dashboard from BeCrop, which is an exciting amount of information. So every circle represents 1 species. The tan are the bacterial species, the green are fungal species, and you get, I'm actually gonna roll out and show you how this works and toggle over to the active. This is the live dashboard of BeCrop. And the one that's up right now is the conventional treatment, and you kind of size it. And these are the species. They're there, all the greens, and you can find out, okay, so right off, I rolled on one and it was Fusarium. Yes.
I just have a question while we're in the BeCrop portal. You know, you mentioned some of the other data that you're collecting and looking at fungal to bacterial ratios, and you know, I'm just curious, like, what brought you to BeCrop in specific? You know, before these awesome tools were on board, you looked at BeCrop, so I'm just curious what kind of led you there versus other tools that you could have used?
Yeah, to me, it's the deep dive. BeCrop is what answers. I could get indicators from other tests, fungal bacterial ratios, and I'll do that. But once I realized that the whole concept of regenerative science was what's going on in the soil. What is that ecosystem? Is it dead or alive? Is it being restored? Is it moving? And BeCrop, to get the DNA results, is absolutely, it's like opening up a window into what's going on below the soil in that ecosystem. And I think as I'll just toggle through a couple of the treatments. This is the conventional, which really surprised me, is the number of species jump back up, which I thought was, but just to look at the dominant fungal species, the greens, there's a Fusarium, this Cladosporium. For the major species, I look them up and you quickly find out what they do. This is just black rot. It rots wood. Articulospora. There's a couple of, that's actually a beneficial fungus there, Epicoccum. And the amazing thing is you can go through and you can toggle on the different phyla, I guess it is. And there's the fungal diversity that I'm seeing. This is the conventional treatment. How many yeast might be there? Just one. Okay. Mycorrhizal fungi, that's my goal, is to get a mycorrhizal fungi population. There's no arbuscular mycorrhiza, no ectomycorrhiza. So bacteria, a lot of species. And this is the new one. Archaea is what they call the estremophiles, and it's a bacteria-like organism. And I'm fascinated to find out that all of these that I happen to have on my farm are all nitrogen fixers. So what a great thing. So that was the conventional, and then I'm going to switch over to the regenerative and zoom out just a little bit. And what you see right away is I'm starting to get fungal diversity. I don't have that big dominant wood rotting fungus. There's probably still, you can look and see what they are.
And just really quick to clarify what we're looking at in this figure, the size of each of these circles representing a species or genus of soil microbes, the size of that circle is proportional to the population size in the soil sample. So when Herb says that the previous sample, the conventional, was dominated by one fungi species, that was the really large circle, and thus large population based on the amount of DNA essentially that we're able to extract on those fungal species.
And so one of my excitements here, so here's my Archaea, which are my nitrogen fixers. And then my goal is some mycorrhizal fungi. So I do have some. Here's an ectomycorrhizal living outside the root. And then I actually have an arbuscular, which is the one that populates and goes inside the root. And I can just roll over it and look it up. And Diversospora, and it has a lot of benefits. So I've looked it up. It adds a lot to a plant's defenses. This is the one that was really shocking to the indigenous. So now you see just a couple of fungal species. They're huge, and but they're not good ones. So we've got a big Alternaria dominance, and then we've got Cladosporium, which is wood rot, and then Epicoccum, which can be a good bacteria. And then this one is Lectera is a grass pathogen. So you can literally see the change. So my indigenous, so adding microbes, this is my conclusion now, adding microbes was a benefit. So I'm going to go back through. This is kind of what we just looked at.
Herb, we just got a really good question that I wanted to stop at while we're,
Sure.
Before we get too much further. We've got about, just really quickly, we've got about 10 minutes left before we want to open up the questions.
Yep.
And so just want to remind the audience that we do have the question poll is open, so please drop your questions. They're just starting to roll in. But one that came up that I thought was good to just bring up right now while we're on looking at all the different microbes, they asked, how can you, in your DNA analysis, how can you tell between what's living and what's dead? And we get this question a lot, so I just want to touch on it really quick while we're looking at this kind of virtual microscope.
Yeah,
so
please.
In the soil, DNA is churning over really quickly. You have bacteria, fungi, larger insects, nematodes, so on, that are consuming other smaller things, and it's just a constant digestive system with a lot of things cycling. So when we take a DNA analysis or we take the soil analysis, getting that soil analysis within a couple of days, we're pretty certain that most of the stuff, 99% of what we are measuring, what DNA we can measure, was alive at the time of sampling. And that's because when we pull a sample and now that biome that we've collected is no longer without the exudates or other food sources, as a microbe dies, it'll immediately get consumed by what else is living, and within a couple of days be unrecognizable on a DNA standpoint. So we can distinguish between what's living and dead DNA, for one, so we can tell the difference. And what we've seen is that most of what we measure was actually living at the time of sampling, because it cycles so quickly. So just wanted to stop
perfect.
The virtual microscope, because I think it helps to.
I love that. I hadn't heard it. Virtual microscope is exactly what it is. I'm going to here's another tool that comes off of the dashboard. I mean, it's just so interesting. So you can pull up by genera or species. So I just put in some of the dominant ones. This one, Articulospora, it's a leaf decay fungi, but look how dominant it is in the conventional versus my regenerative. Doesn't have that. A couple of ones we looked at, just the level of alternaria. It's the comparison that I go in and make. I love these dashboards and to play with them. Fusarium, real negative. So 5% of my fungal population was still Fusarium in the regenerative, but it's 15%. So I got a threefold increase in Fusarium in the conventional. Trichoderma, real beneficial fungi. Actually can consume and protect against other fungi. So I've got 10 times the level of trichoderma in my regenerative that the conventional has. Just a couple of few. These are archaea that I'm so fascinated by, my nitrogen fixers. So nice little bump up in the level when I put all the archaea together in a graph.
There was another, before we hop into the indexes, which are really awesome, just another great question on species diversity and what's beneficial versus might be pathogenic. So the question was, within the context of regenerative philosophy, how might our understanding of microorganisms shift based on, what we call beneficial versus what's not beneficial, and the biology is really complex, right? So the way
it's a great question, and it's probably brand new science. So some of the brilliant people in regenerative agriculture, Don Huber and some of the others, are identifying that some of these fungal pathogens that we know about, Alternaria, Fusarium, are endophytes. They go live in the plant, but under certain conditions, they can actually have a beneficial effect. Of course, as a plant pathologist, I never thought of it that way. Any fungi was a bad one. And now I'm realizing that probably 80 or 90% of them are beneficial. Now, and we're just learning how some of them can actually shift based on the environment. If a plant is weak, then they're taking advantage of it. If the plant is strong, sometimes they actually are supplying nutrients. So that's quite a new area of science.
And the way that I kind of think about it is that there are kind of the broader categories of beneficials, the communities that we need to be doing the things that we need to happen. Like on this next slide here, we're looking at different hormone production and nutrient mobilization and so on. So those are like our key beneficials. It's kind of like in a grassland system, you have your cornerstone species, your best, most palatable grasses that the animals want to eat. And they're really like ecosystem indicators, right? So you have your key beneficial families, And then you have this large range of opportunistic organisms that could be beneficial. If they get overpopulated or there's too many of the same type, then they could become pathogenic. But there's a large category of opportunistic bacteria and fungi that are only really bad if they're not kept in check. And that's one of the importances of like bacterial fungal ratios and so on. And then there's a very small subset of those that are just truly pathogenic that we want to keep an eye on, or are maybe concerns if they're over a certain abundance or so on. But what's really important when we're talking about disease and what organisms are good or bad is that that's not, like Herb said, it's not the only portion of the disease triangle, right? What nutrients are available to the plant to help them thrive and be healthy is really important. What beneficial organisms they have to promote stress adaptation is really important. Supporting plant health overall is just as important, if not more important, than whether or not we have pathogen abundance.
Yeah, and I mean, that's a great explanation. And this science is, to be honest, so new. And I think that I'm seeing it in my trees. I don't see anything pathogenic. People around me are spraying for insecticides. I've got one insect that gives me a hard time. Virtually no weeds because of my cover crops. And so I believe I'm getting a healthier plant. I've got this entire new ecosystem going on underneath. The beneficial guys are taking over. And so here's this heat map, just to kind of leave some time for questions. This is the roll up that BeCrop gives you, which is really helpful. So they know BeCrop now has 14 million microbial species they've identified and are linking the function of each of those. So when they roll them together, so there's probably 20, 30, 40 different species, and then they've got the quantity of each one that go into each of these measurements. So I've got bumps in exopolysaccharide production, heavy metal solubilization, salicylic acid, which is great. That's disease protection, systemic acquired resistance, sialic acid, which has a lot of growth hormonal effects. I got a nice bump in zinc, as they can measure, which is for citrus, it's leaf size, carbon increase, and then a drop in chlorine, which is actually a positive thing. So it's kind of a good roll up that's available. I wanted to, this is important for me to admit here at this point, that these are the growth. So I take tree caliper measurements on every tree in this trial, at least 3 times a year. What happened was right at the beginning, when the trees got their first shot of synthetic fertilizer, high nitrogen fertilizer, the conventional trees jumped out ahead and almost doubled in size. And, it was quite a discouragement that first year. And the regenerative and the indigenous are growing at a constant, steady rate. And so that's the decision I'm making. I'm choosing to grow the healthiest tree I can grow. It's going to eventually give me nutrient dense fruit that I can even market in a completely different market. So I did give up some immediate big growth on trees right at the beginning, and actually some regenerative people had cautioned me. They said, it's going to be slow to start, and it's a choice I made. But I love right here at the end, the regenerative is starting to angle up and grow at a higher rate than either the indigenous or the conventional trees.
Yeah, I love that, because it's really highlighting, just an ecosystem, maybe even back to the conversation we kind of started around cover cropping and competition. And a lot of what I've heard regarding competition of cover crops is that, in year 1 or 2, it can appear like there might be some competition, right? Because there's, initial competition for nutrients, regrowth, water, and so on. But as the cover crops and the other practices that we're implementing start to really drive down and create a healthier ecosystem overall, each individual in that ecosystem has a greater opportunity to thrive. So this is really cool, and I'm excited to see, as you continue tracking this information, where it goes and what the size of the trees and the yield is relative to the conventional side as well. So it's great.
And Marie, I'm going to show you, this is it right here. So this is the simplest measurement that anybody can take. So there's a metal probe called a penetrometer, and you put 200-pound PSI pressure on it. As soon as I get 5 feet away from my tree, I can only get that probe 3 inches into the ground with the 200 pounds of pressure. When I go between the trees or anywhere close to the trees where I've got cover crops, microbes, and mulch, that penetrometer, sometimes it sinks all the way to the handles. And I first noticed this when I'm walking through the grove. And you can see how sad the trees are. They've lost their leaves from the winter freeze. But as I walked perpendicular across the rows, every time you got to a row, the ground raised up, and it was 2 to 3 inches higher, and the ground underneath my feet was spongy. And that's the most exciting indicator that I've got. I am got a population under there it's root mass, it's cover crops, it's tree roots, and it's microbes. And they're all interacting, and my soil is literally becoming porous. I've got rain gauges all throughout, soil probes that are soil moisture probes, and you can literally have them different distances out from the trees, and you can see the ones closest to the trees are right in the row. Incredible the amount of porosity that the rainwater or irrigation goes right in. So this was an exciting confirmation of everything that I've been doing.
And Herb, just to tie in something from the BeCrop report that we saw, that exopolysaccharide measurement you mentioned, exopolysaccharides are a biofilm type substance that's effective at basically gluing the soil together, improving soil structure, porosity. So that right here, it's really exciting to see that these measurements you're taking in the field are kind of confirming what we're able to see in the BeCrop. The BeCrop is showing kind of the driving factor behind what you're seeing in the field.
Absolutely. That's the beauty to me of doing all these together. You got the above ground, the visual, the physical measurements, and then you've got genetics. Well,
Herb,
everyone wants to know what happened in our freeze.
Well, Herb, actually, can you go back one slide?
Sure.
We have so many questions, and we only have about 5 minutes left, and there's just some really amazing questions here on what's going on. And one of them has to do with this last photo. So I just wanted to pull us back there. Just a really quick question on what the black container around is on the base of the trees and what that is.
That's unique to citrus. It's called a tree teepee. And basically, it just gives, the sprinkler is inside that teepee. And so it projects the warm water up around the tree when you need frost protection. To be honest, that's its biggest function. And I also reduce competition right immediately for about a foot around the tree. Yeah. I only have them on about half of my trees. I put them there for frost protection.
Great. Okay, that's awesome. If you want to touch on the freeze really quickly, we've got there's like 20 questions. So
That's great. This is what happens, and this is what it looks like. It froze for 5 days straight at Christmas time, and we turn on the sprinkler, and it doesn't freeze the trees. It's not like strawberry. You just spread it. You let the warm air waft up across the tree, and it keeps the graft and the scion. Every citrus tree is grafted. And so I thought I'd go farther and I'd do the old fashioned. I grew up in Florida and they started fires. So I had 50 fires going in my grove, and I don't know how much it helped, but anyway, that's what it looks like. It's quite a shock when you get out there the next morning. I stayed up all night long lighting fires. But here's some of my questions, and please, Marie, just go ahead if you gotta pick some of the ones that.
Yeah, I'm gonna pick a couple here, and I'll ping some to Gus too. Some questions are specific to BeCrop. So maybe Gus, you can elaborate really quickly on, just talk about the stress adaptation section of the report. Herb pointed out a couple pieces on salicylic acid and abscisic acid. Can you just talk a little bit more about what we're looking at when we look at stress adaptation on the BeCrop?
Yeah, sure. And I think in the context of citrus, those are some really interesting microbial functions to look at. So when Marie says stress adaptation functions, that's a section on our report that looks at different phytohormones and other compounds that microbes can produce and essentially donate to the plants. And these are all compounds and phytohormones that help the plant deal with stress. So it includes salicylic acid, abscisic acid, Herb mentioned, exopolysaccharides, salt, metabolites involved in salt tolerance. So what they all have in common is they help a plant continue to grow and thrive under some form of stress, or help a plant activate responses to pathogens or disease. And in the context of citrus, with citrus greening taking such a massive toll across a lot of the Florida industry, these are really important because we don't know, currently there's not an established way of curing or eradicating citrus greening once it's established in a grove, but we have seen that trees can continue to produce economically viable yields if they're well taken care of. And those stress adapters that we measure with BeCrop are very important to ensuring that a citrus tree that's infected by greening or canker or another disease is able to continue to produce despite that disease, and that the soil biology is doing everything it can to support that plant and minimize the impacts of the disease and susceptibility to other diseases too.
Yeah.
Gus brings up, that's a real important point. In Georgia, we do not have the vector for the greening, HLB greening disease. So we probably will one day. And my hope is that my regenerative trees will have a lot better chance of defending themselves against greening. It's reduced Florida production by 70%. It's could potentially wipe out the industry there. So regenerative is a solution, and people are finding that. The few regenerative growers down there are having a lot better luck recuperating trees.
Oh, this is an interesting question. So the question is, one of the reasons that I hear from growers keeping their alleyways bare is that it helps protect them from frost. Now, interestingly, I hear this everyone in different regions has a different reason for keeping the ground bare. That's a new one. I work in a lot in California. Anyways, the thought is that the black soils give off more heat, but I understand. So I guess just what's the truth there?
It's true, and it works for about 4 hours. And we had below freezing for 48 hours. So yeah, for the first, you get the soil wet before the freeze comes, and the warm water stays for quite a while. But after 6 or 8 hours, that bare soil is a detriment, and you're going to have a lot worse damage. So
right.
Yeah.
I guess the overarching theme here that I like to think about is that anytime we have ground cover, anytime we have a more diverse microbial community, we have a buffer. You have a shorter temperature variation or a smaller temperature variation if you have ground cover. Your black top, right, your black bare soil may collect more heat during the day. It's going to get hotter, which
Right.
on a hot day is not going to serve you. But on a, when you're getting cold, it might help to hold more heat in, but it's going to lose that heat very quickly because it's bare. So while it may be warmer in one instance, it will be, cooler. It'll actually get colder as the longer time goes on, like Herb said. And not only does it buffer temperature well, like ground cover and biology buffer temperature, it also buffers water infiltration and water holding capacity and pH. And so all of these things, when you have more diversity, when you have ground cover, when you have diversity in your species above ground and below ground, you have a greater buffer and greater resilience overall. So
it's a lot of work to manage, but I like the ground cover right up to the tree. I really think that my microbial life gains from it, and eventually that'll be the tree. And I want to, right here at the end, as we finish, here's my dream, is that for this entire regenerative industry, that one day we will be able to document and prove to the public that food is medicine, and that if it's grown correctly, it's going to be nutritious, and it's going to resolve a lot of the terrible endemic medical problems that we have. So that's my dream, to get there one day.
Well, I think that's a great way to wrap up, Herb. Thank you so much for joining us. There was a ton of other questions, so Gus, Herb, and I will get back to everyone that has questions.
That'd be great.
Sorry we weren't able to get to all of them today, but we're at the top of the hour and we want to respect everyone's time. But this was really great discussion. Amazing work that you're doing, Herb. We really appreciate you being here and joining us and putting this together and just sharing with the industry, continuing to share your expertise and the amazing research that you're doing now on your own farm.
Well, thanks to Biome Makers too. Y'all are doing something amazing. Yeah.
Thank you. Yeah, the way that I think about it, just like to your point around food as medicine, is like what data we provide is validating what we can all feel, right? What we all know is true. When we look into an ecosystem, the ground is covered up to the tree. Nature wants it that way. So it's what we know and feel intuitively, I think, makes the most sense. And now it's just putting the data behind it because that's what we need. So, but yeah, again, appreciate you, and we'll see everybody on the other side. Thanks everybody for being here.
Thanks everybody for joining. Yeah, it's been awesome. Thank you.
Thank you.